Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Biology of SARS-CoV-2, Past, Present, and Future. My name is Sonia Hunt, and it's my pleasure to be your X-Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout this presentation using the questions chat box, and we'll try to attend to your questions during the presentation or at the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank GenScript who developed the content for this presentation. GenScript is the leading contract research organization in the world, providing gene, peptide, protein, CRISPR, and antibody services. Since its foundation in 2002, GenScript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. The company is recognized as having built a best-in-class capacity and capability for biological research services, encompassing gene synthesis, peptide synthesis, custom antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. Now it's my pleasure to introduce you to your speaker for today's webinar, and he is Dr. Benjamin Tanover, Professor of Microbiology and the Director of the Virus Research Institute at New York University. Benjamin completed his postdoctoral training in virology from Harvard after receiving his PhD in medicine from McGill. He is presently a professor of microbiology and the director of the Virus Research Institute at New York University. He is a Fulbright Scholar and the recipient of a Presidential Award in Science and Engineering. Benjamin's research program focuses on the interactions between viruses and their hosts, with a specific emphasis on SARS-CoV-2 in response to the COVID-19 pandemic. His lab is supported by the National Institute of Health, the Department of Defense, and the World Health Organization. Now it's my pleasure to pass over the mic over to Benjamin. So Benjamin, when you're ready, you may begin.
Well, thank you very much, Sonia, and thank you to all of you who are attending this webinar. I certainly am looking forward to a day where we can do these events in person, but this is one of the silver linings of the pandemic, in that at least when you give a seminar, really there are no boundaries or time zones that stop you from being able to attend such an event. And in that light, I hope that this is a seminar that you find enjoyable, and as Sonia said so kindly, please feel free to interact. Probably the more you interact with me and the more questions you provide via the chat, the better this talk will probably go. So with that, I was given the task by GenScript, who has really been very supportive in our research here, to give this talk on the past, present, and future of SARS-CoV-2. And obviously that's quite a feat to do in 45 minutes. So I'm going to give you a taste of all these things. And like I said, please feel free to ask any questions should anything require more detail or you want to know more about something. Since the pandemic began, actually prior to the pandemic, I was working on influenza, and prior to that on a variety of other different viruses. And so my research is always kind of focused on how host and virus respond to each other. And of course, because of the pandemic, our lab switched to focusing on SARS-CoV-2 almost exclusively for the past couple of years. And I've been using this title slide ever since that began, and just watching these numbers climb. And as of yesterday, we hit the 400 million mark of number of confirmed cases as the deaths approach 6 million. So clearly, a life-changing event for all of us. Next slide. And so just to give us all a sense of at least the past, the first part of our title was, I'm sure as many of you are well aware, in 2019, really at the end during kind of the winter break for most of us, there was this gurgling of the media about a virus of unknown origin, or maybe a bacteria of unknown origin that was coming out of China, and it was quite clear that it was somewhat concerning. But the truth was that it wasn't the first time that events like that had happened and then fizzled out into nothing. And so it probably wasn't until early January where we learned that actually this the agent underlying this unknown pneumonia was actually a very close cousin of the virus that we saw in 2003, and that is most of us in the lexicon just refer to it as a SARS, which is part of a much larger family called beta coronaviruses, and this one was a very close relative called SARS-CoV-2. And really what made SARS-CoV-2 different than the SARS from 2003 was really just its external protein called spike, so the attachment protein that a virus needs to be able to engage a cell and get into that cell. It looked like the entire genomic structure of the virus was almost identical to the 2003 strain, except that the spike protein had changed dramatically, and the closest relative to that spike protein was another beta coronavirus that came out of pangolins, which are kind of this weird ant-eating look-like animal. And so this theory was born that perhaps an animal got infected with two different coronaviruses, and this kind of hybrid of the two emerged out of that. But since that time, I think we've come to realize that there are actually population of bats out there that have SARS-CoV-2 relatives that are very close to its original sequence, and so it probably just came from a bat. In any case, it slowly migrated through Asia. It went into Europe, and actually, I think it was the impact it had on Italy that made it very clear that no amount of climate or weather or genetics were ever going to stop this, and that we were all in for a pandemic. And indeed, in March, the World Health Organization declared it as a pandemic, and at that moment in time, we already had a pretty good sense of what mortality was going to look like. It obviously skewed towards the elderly and those with comorbidities, which is very reminiscent of influenza. And one of the perhaps saving graces of SARS-CoV-2 was that at least it spared children. And that's probably because children in general have less of this ACE2, this angiotensin II converting enzyme, which is the target of that spike protein that allows it to get into cells. And so because especially kids prior to puberty have less ACE2, it's more difficult for the virus to infect and cause disease in them. And so for that reason, we have this very strange skewing of mortality. Next slide. All right, so this is the beast that we're all talking about. This is how the genome of the virus looks. I'm unaware of the overall makeup of my audience here, so I'm going to talk in relatively less scientific terms wherever I can. And so this virus this is the genetic material it's made out of, and it's camouflaged to look very much like the same kind of material our cells make, something called messenger RNA. And so as soon as this RNA structure gets into your cells, it immediately engages the host cell machinery to start making some of its own proteins. So the first thing it does is it creates, we call it a polyprotein. So where you see ORF1A, that stands for Open Reading Frame 1A. And what you're looking at there is it's simply going to translate this mRNA into a protein, and that protein will get cut up by a virus protease, like a pair of scissors that will cut up the protein into many pieces. And all of those pieces are going to assemble together to make a machine that can use RNA to make more RNA. And that's not a machine that humans have, but it's a machine that obviously RNA viruses need if you're going to live in this world of RNA only kind of space. Then the virus has another trick up its sleeve. It has many tricks. The next trick it does is you can see there's a kind of another box right beside ORF1A called ORF1B, but it's offset a little bit and it's changed in color, and that's because those instructions have a small mistake where the blue and the brown lines meet together. And it's not really a mistake, but if you were to look at it from, from a very high level, it would seem like the virus made an error. But what it's actually doing is it's capitalizing on the fact that that error will be incorrectly processed, say, 1 in every 100 times it gets processed by our host cells. And so it's a way for the virus to make more protein from that ORF1B frame than it's going to need. And so this virus does this trick, so it makes a lot of ORF1A, but a lot less of ORF1B because it needs a lot less of ORF1B. Ultimately, though, these two polyproteins get cleaved into this massive RNA making machine, And then the real biology begins, and everything that happens downstream of that, that S is for spike, that's the one I was just mentioning, we'll see that in a minute, as well as all these other accessory genes behind that. They are really there are a variety of structural components and components that are going to interact with the cell, but it's actually the biology of the virus concerning those genes that I really want to talk about next. So we go to the next slide. So I mentioned that ORF1A and ORF1B, this now red and blue boxes you see, they're going to become this massive machine called an RNA-dependent RNA polymerase. It's just a huge complex that can take RNA and make new RNA using RNA as substrate, which is something, like I said, we don't do. But after it's made that machine, this virus performs this really extraordinary trick. And it's really, I think, this trick that separates SARS-CoV-2 biology from, say, influenza, and what makes it more serious to us. And so what it's going to do is it's going to make a miniature version of a genome that's much, much smaller than this massive 30,000 nucleotide genome. And to do that, it's going to start replicating, pretending it's replicating. So it's going to make a new strand. So it's going to bind where it says 3' there on the end of the genome. And it's going to start making a new complementary strand at that 29,674 position. It's going to start moving back towards the 5' end to make a genome. And if, without my pointer here, if you just look down where it says these two TRS elements are really close together, that stands for a transcriptional regulatory sequence. And the main take-home message here is that when this machine, this RNA-dependent RNA polymerase gets to a TRS element, it needs to jump essentially from one strand of the genome to get to the other strand of the genome. And it needs to do this because in order for that machine to recognize RNA as viral RNA, it needs to have the exact same ends as the genome in the beginning had, that same 5' and 3' sequence. And so in order for the virus to do this, it makes smaller genomes that are mostly comprised of what's on the 3' end of the genome, but they're adding on a little piece of the 5' end. And because the machine has to do these acrobatics on the RNA, it results in the production of a lot of pieces of RNA that are going to fall off, are going to be broken, are going to be two pieces of RNA stuck together. And all of those things, all of this aberrant RNA are all the trigger points for the way we, our cells anyways, recognize virus and elicit a response to that virus. And so what's interesting about this is that, to put it in maybe more colloquial terms, is that this virus is very sloppy. It makes a mess. I mean, I have two little kids at home, and if you gave them carte blanche in a room full of toys or really anything, if you gave them a couple of hours, they would create massive chaos in that room like any average kid would, right? And I would think that SARS-CoV-2 is very similar in that regard. It's taking over the cell, and you're going to see in a minute, it really does take over the cell. So it doesn't really care that it's making a mess because it's about to destroy the cell anyways, but it's the mess that it makes that creates all the consequences downstream that cause us disease. And so that's why we're talking about it. And this is really the underlying reason why. It's because it has this funny biology where this RNA-dependent RNA polymerase is forced to jump from one end to the other end. Next slide. All right, and so you can see that. So this is using an antibody that recognizes two pieces of RNA stuck together, or double-stranded RNA. And so those RNAs I was talking about where the jumping is necessary, and I was equating it to the virus making a big mess, we call those subgenomic RNAs. So that little S, little g RNA, a subgenomic RNA is the shorthand for all of those products. And what you're looking at here is an antibody that recognizes when you have this double-stranded RNA present. And now our cells should never have double-stranded RNA. Double-stranded RNA is a universal sign for a virus. And that is for the same reason we don't have a machine that makes RNA from RNAs, because we use DNA as our, essentially our hard drive to run programs. Whereas RNA viruses have to live in an RNA world where they only go from RNA to a different type of RNA. And so they make this machine. And as a result is when you're replicating and making more of yourself, you're always going to have intermediary products that have two pieces of RNA in very, very close proximity to each other that can form helical structures very much like what our DNA does, but as two pieces of RNA. And since that's something you and I would never produce, we have evolved to be able to recognize that structure and elicit a response to it. And so this antibody is looking to the presence of that. And so in an uninfected cell in mock on the left here, you see absolutely no green signal. And on the right, you see a lot of green signal showing that this virus is making a very large mess of every cell that it infects. I do see a question here, but it's one I'm going to get to later on anyway, so I'm going to hold off onto that one. but please keep your questions coming. I like them. Okay, next slide. Okay, so what do we do in response to this? Okay, so this is going to be a very high level, but to the left here, this is my cartoon version of SARS coming in. So that green shaped thing on the outside, the studded circle, that is the spike protein, or S gene, that is going to allow this virus to get into a cell. And so it's going to bind something on the surface of your cells, which is, of course, this angiotensin converting enzyme 2, or ACE2. And so cells that have ACE2 are naturally susceptible to this virus. And so it's going to bind that ACE2 receptor. The binding of that receptor is going to trigger a process where the cell brings in stuff from the outside. The virus is going to escape that process and get into the cytoplasm. Excuse me, sorry. And it's going to start translating. If you remember, we're going to translate that ORF1A and that ORF1B to make our machine that takes RNA and makes more RNA from it. And so if you kind of follow this kidney-shaped product down, these are just kind of very simple cartoon depictions of this virus that's replicating. But it's generating, you can see a purple squiggly line and an aqua squiggly line. This is my attempt to depict double-stranded RNA in the cell. And so there are proteins that just hang out in your cells, in all of your cells ubiquitously, that are looking for the very production of this kind of double-stranded RNA. And so two good examples of this that were recently discovered, 5 or 6 years ago now, are RIG-I and MDA5. And the names don't matter, of course, but what they're doing is when they can sense the production of double-stranded RNA, that equates to that cell that there's a virus present, and then that virus is likely replicating because it's creating this double-stranded RNA intermediate. And as a result, it goes through this, it basically engages a program, it turns a program on through a number of transcription factors and kinases, and all of this machinery is very well known, but we're not going to get into those kind of weeds in this talk. And ultimately, we're going to land on these two major activators. So they're written here as IRF3 and NF-kappa B. These are just, these are things that can turn your genes on or off. And in this particular sense, the way this pathway works is when your virus, or when your cell sees a virus, it's going to flip these switches so that IRF3 and NF-kappa B both turn on, and they work together to initiate this antiviral response. And the way that works in general is that they elicit the secretion of a protein called interferon. And interferon, there's different types of interferon, we won't need to get into that, but it basically gets secreted, and it is a call to all the neighboring cells that says, hey, I detected double-stranded RNA in my cell. I think there's a virus here, so I'm secreting interferon so that all the surrounding cells can be aware that there's a virus, and they can fortify their defenses before the virus gets there. And that's exactly what happens. And so the interferon gets released, it binds to all the neighboring cells, it sets on another set of programs that culminate in the production of about anywhere from 200 to 300 different genetic genes that are all aimed at stopping virus infection through a whole variety of processes. And this is like how we have evolved as our first line of defense against virus infection. Next slide. And so in order for you to be a virus, you really have to be able to navigate this process. And so to do so, basically every virus you've ever heard of that causes disease has to be able to shut down and block this. And so one of the things we do initially when a virus like this emerges is we try to figure out what this virus is doing that's allowing it to get past that interferon defense. Because if you can figure that out, often you can find drug targets or ways that you might be able to treat it. And so this is a relatively fancy way to do that. But what you're looking at here is something called single cell sequencing, where we simply look at infected cells in a dish, we throw virus onto them, but we throw virus onto the cells at something we call a low multiplicative infection, meaning there's many, many more cells than there are viruses. So the virus is going to have to work in order for this to really infect all of these cells. And then before we give it the time for this virus to infect all these cells, we actually stop the experiment and we take a snapshot of everything that's going on in that cell at that moment in time to get a sense of what is the virus doing to the cell and what's happening to all the cells around those infected cells, initially just to get a sense of, like, this would be the very first couple of cells that get infected in, say, your respiratory tract. And so, what we're looking at here on to the left here, every dot is a cell, and the orange dots are cells that are infected with virus. And we know they're infected with virus because the snapshot that we took was a snapshot of all the RNA in the cell. And so this little heat map you're looking on the right, where there's a big block of red, those are all the genes. So red just means that you detected them on their way up. you can detect all of the components that make the virus in those cells, and that's why we know they're infected. And so in the orange cells, they're very much infected, and the cells that are purple, these are cells that are either just beginning to be infected or they're still uninfected, but they can respond to the signals that are being induced by the infected. And so in that way, you look at the snapshot of things that aren't viral genes, and what you find is kind of interesting, because normally what a virus does is it just shuts down those two switches, or if you remember IRF3 and NF-kappa B, it switches them off in a variety of different ways so that this initial defense can't ever get started. And one of the quirky things about SARS-CoV-2 is that it leaves one of those switches on. It turns one switch off, but the other one stays on, which is already quite unusual. So NF-kappa B stays on. So amongst the orange cells is one of the only things that's happening is that you have some limited host activity going on as well, and we'll talk about that in 1 minute. And then in the purple cells, this is much less surprising, but what you're seeing is that interferon. So you remember that warning signal of, we see double-stranded RNA, we're infected by a virus, get ready, fortify. That's happening in all the cells around us in this particular culture dish, and we'll talk about that in more detail in a bit. Next slide. All right, so we're going to just take a slightly deeper dive into what's happening in those cells. And so on the left here, this is a truly remarkable graph that's probably lost on most people who don't eat and breathe virus biology every day. But if you take the best strand of influenza in a given winter that was causing a lot of disease and a lot of snotty noses, and you were to do this kind of experiment with them, if you looked in the cells that were infected with influenza, and took a snapshot and said, okay, let's say if you think of a cell as a computer, for example, and there's so much processing power, the bandwidth that influenza would take having infected that computer or that cell is at best maybe 5%, meaning about 5% of the running memory is being dedicated to the virus, whereas 95% of the activity is still the computer doing its thing or still the cell doing its thing, depending on the analogy you want to use. And what SARS-CoV-2 does, though, is remarkable in that it goes well beyond 5% bandwidth and can achieve everywhere anything over 60 to 80% bandwidth, meaning that at the moment in that 24-hour time period there where you see that bar graph go well above 70% of viral RNA, that means that we took a snapshot of these cells that were infected, and those cells were more virus than they were human at the time of infection, because the virus not only is very good at making more viral products, which is what we see in the middle there, which I'll explain in a second, but it's also very good at getting rid of the host products. And so what's happening there is, again, this is still single cell sequencing, but we're taking a snapshot, remember, of all the RNA, and we're trying to figure out what's going on. And so the yellow there, those are the new virus-specific genes that are coming up. So they don't exist in mock because they weren't infected, right? But the blue line should still go all the way to the top, except that we're not identifying all of the pieces of a cell that should be there because the virus is actively destroying them. And then on the right, to that same point, is the only blue that's remaining, meaning that since the virus is taking up so much of the bandwidth and leaving so little for the host to function, the only thing that really is functioning, and this is data where we look at the genomic DNA and we look to see what's still on, is what we see is that NF-kappa B, again, remember we had that oddity, the NF-kappa B stays on. And there's a consequence to this, so that in infected cells, it's mostly virus with the exception of this one biology, and this one biology happens to be very much in charge of a different aspect of the defense, which we'll talk about next. Next slide. And so I see a question here from the audience about how do we know about like relative abundance when we look at single cells, and that's actually quite easy, because the overall numbers of a given cell, when you sequence a cell, the snapshots that you take, you're going to get something like 100 million reads or pictures of individual pieces of RNA. And so you can do everything as a percent of total. So, if you want to look at gene X, you can say that gene X is, 0.001% of all the components in a given cell. And so it's very quantitative to compare from one cell to the other cell. But good question. Okay, so back to our story at hand. You remember we turned off, we saw that NF-kappa B was on. We saw that the host proteins were being destroyed and the virus was essentially taking over. And we learned early on that the virus is a bit of a slob. And so this is, again, kind of taking that same high level, but getting a sense of like where the virus is actually blocking. And so you can see that IRF3 there is being attacked by 3 different products, a product called N, a product called ORF3B, and a product called PLP, which is actually a protease. And it's really investing a lot of energy in making sure that IRF3 switch doesn't work anymore. And that makes a lot of sense because in the absence of that switch, you're not going to get the interferon output that the cell is going to want to elicit. And remember, that's the call to arms, right? That's this telling everybody to fortify and get ready. So if you can minimize that, that's definitely beneficial for the virus. And so it's not unusual at all that viruses do this. It's just unusual that the virus does it by targeting only IRF3, but leaving NF-kappa B on. That part's a bit quirky. And then on the right-hand side, this is for the cells where maybe they saw interferon before they got infected. Even in those cells, this virus has a number of antagonists in its bag of tricks, one of them being this NSP1, which kind of destroys all the host RNA. So we saw that in the single cell sequencing. And they also do tricks like they prevent some of these much needed switches, these transcription factors, from going into the nucleus where the genes are to turn on. So if you can stop that process, you can also stop the cell from eliciting any kind of defenses. So this is kind of like the virus takeover at a cellular level. Next slide. Okay, so I wanted to take you one step higher to help you understand the consequences of NF-kappa B. So this is still the same thing we were looking at. I'm just taking away some of the graphics. And so if you remember, the very first thing that happened was we made double-stranded RNA. So in the business, we refer to these as PAMPs, or pathogen-associated molecular patterns. They are things that viruses or pathogens make that we don't ever make as humans. And as a result of that, we have evolved to recognize those structures as something that we should elicit a response to. And so those things that make that recognition, we call them pattern recognition receptors. That was the RIG-I and MDA5 I mentioned briefly. Then those guys go through a variety of pathways to turn these switches on. And those switches are controlled by things like transcription factors, which are NF-kappa B and IRFs. And then those elicit a cellular response to achieve what we're trying to achieve. And as it relates to viruses, we're really trying to achieve two things. And so first to the right, so as I mentioned earlier, is this call to arms. So that is, remember, NF-kappa B and IRF3, we switch them on, the cell releases interferon, and it warns all the neighboring cells, a virus is coming, fortify, get ready, right? That is a very important part of this immune response, but complementing that part is the left pathway, which is mediated by NF-kappa B. And that is more a call for reinforcements. And that is to say that, you know, if this is happening in your lung or in some tissue and the virus is taking a foothold and replicating, it's very often that the call to arms is not sufficient enough to actually kill the virus or neutralize the virus. It's really only there to do its best to slow the virus down. In order to actually resolve the infection, you need some professional immune help. And that's where the call for reinforcements comes in. And so NF-kappa B is a major driver of that. So it sends out basically a warning signal in this form of other secreted proteins called chemokines. And it's going to create almost a yellow brick road trail where the most concentrated yellow is going to be at the site of infection. And the farther away you go from it, the yellow fades and fades and fades. And so as cells that are going through the blood, like T cells and B cells and monocytes, are able to see this trail, they follow it until they get to the end of the trail where it's the brightest, and then they find the pathogen that they then need to deal with. And that's exactly what's going to happen in the case of SARS-CoV-2. We're going to bring in all these immune cells to the lung. And so if you click on one more advancement, what you find is because this virus stops the IRFs, we can block a major part of the call to arms, but we're going to leave the call for reinforcements on full blast. And that dynamic is basically the underlying biology of COVID. All right, next slide. All right, so how does this all connect? Why am I even talking about this in this way? Let's go to the next one. So we're going to really get into hamsters here, okay? And so I don't in 45 minutes really have the time to convince you that the hamster is really the perfect model for all things SARS-CoV-2, but there are lots and lots of publications out there that validate it. And I'm going to show you that I think I have yet to see any aspect of COVID disease being unable to be replicated in the SARS-CoV-2 hamster model. And this is totally stochastic, of course. This is really just has to do with the fact that the hamster's protein sequence for its ACE2 receptor just happens to be more compatible with this virus and more human-like in this one respect than our normal go-to small animal models like mice. And so if you wanted to use mice in SARS-CoV-2, you either have to adapt the mouse to have a more human-like ACE2, or you have to adapt the virus to be able to recognize the mouse version of ACE2. And both of those things come with consequences, and it kind of changes the biology between host and virus. And for that reason, we really stick with the hamster model. Next slide. All right, so one of the early studies, really great study that came out of kind of a joint lab that exists both in Japan and in the US here from Yoshi Kawaoka's group, showed that if you look at the lungs of SARS-CoV-2 infected mice, you can see something that's often referred to as this ground glass opacity. And basically what you're looking at here is the fact that your lungs are taking on fluid, which makes it much more difficult to breathe, which of course is the ARS of SARS, is the respiratory distress syndrome, the acute respiratory syndrome. And what you're looking at here is basically that in a mock lung, you see a nice healthy see-through lung, but in a SARS-CoV-2 infected lung, you're now seeing these essentially like clouds forming inside the lungs that look a lot like SARS-CoV-2 people when they're hospitalized, which is what you see on the left there. So just to say that it's phenocopying, we call it the disease. It looks very much the same. Next slide. And so, we also got in very much into the hamster model, and of course, hamsters are not widely used in the lab for most viral studies. So this was a learning curve for most of us. And so to the left this is just kind of characterizing the model, but I put it in here because I wanted the audience to appreciate how consistent this is and how much we can leverage this animal model to learn more about how to treat this disease and what causes this disease, and even get into long COVID at the end. And so SGN, if you now remember, is the subgenomic version of N. So it's, remember, that did that crazy acrobatic loop of the polymerase on the RNA. And we're just giving a virus in the nose to hamsters at increasing concentration. So in the first column, we give them just 10 infectious viruses, very, very little, then 100, 1,000, 10,000, 100,000, and so on and so forth. And all I want you to appreciate is that in all cases, the animal gets infected and propagates that infection. In fact, the more you add, you actually see that the overall levels go down a little bit. That is a strange reality of the fact that viruses, being as sloppy as they are, start making broken versions of themselves, and that's what causes that phenomenon. And then to the right, I want you just to appreciate that if we look at this by plaque forming units, the one in the middle there, yeah, that is a really good measurement of. So when we want to know if virus is present in a tissue, we can take a snapshot of its RNA, like we've been talking about, which doesn't really tell you if the virus is replicating or infectious. It just tells you that it was there or pieces of it are there. This is an assay where we're going to take liquid from the tissue of interest that we're talking about, and we're going to put it onto other cells, and we're going to see if it can establish an infection in there, which would tell you it's infectious. And you can appreciate that, 3 days after infection, every single hamster has 10 to the 6 individual virions per milliliter of homogenate that we have. So they're very, very high levels, and you can see every one of those dots is a hamster, and you can see the fact that they're all clustered together. That's how consistent this is. And this, obviously you can do this by RNA, which is the one on the right. Next slide. Yes, there's a question about long COVID. We're going to get there. I should probably speed up a little bit. All right, and so just to say that, you don't have to artificially put it in their nose to cause an infection. If you give them infected bedding, they will get infected. So this is, we call this fomite transmission. If you give one hamster SARS-CoV-2 and then put in a naive hamster, it will get SARS-CoV-2. If you put a drop of virus in the eye, the infection will catch in the lung, or you can do it intranasally. And again, all of this is true for humans as well, of course. Next slide. All right, and so what's actually happening is, so this is, we call this immunohistochemistry. So it's the staining of the lungs. So you're looking at really big airways here. So those big spaces is where every time you inhale, you're breathing the air through those large tubes, and they're going into finer and finer tubes. But this is where you have your airway exchange, which happens to be those cells that are expressing ACE2. And so the carbon dioxide oxygen exchange is obviously critically of importance, but it's also the first thing that's going to get hit by SARS-CoV-2 virus. And so on day 2, you start to see these little brown spots right in that large area. Those are individual cells, and the brown is staining of the nucleoprotein. So this is the appearance of virus. And I hope you can appreciate that those few individual cells on day 2 give birth to all cells on day 4, where the entire large bronchi is very, very infected, and the virus is most definitely winning on day 4. And then on day 6, you've kind of exhausted your resources. You've killed a lot of your cells that had ACE2, and so you're forced to go further and further into the tissue to find cells to continue replicating. And so at day 6 and day 8, you see it's getting more and more diffuse, but at this moment in time, you will see that you start making antibodies, you start fending off the virus, and by day 14, all the virus is gone, but ironically, there is still plenty of material left. And we are again going to get back to this phenomenon, as I think there is quite a bit of significance. And this again has to do with the fact that the virus overall is quite a messy slob of a virus. And so it leaves so much material behind for the host to clean up that it comes with consequences. Next slide. There's a question here about whether I think that the subgenomic N relative expression might be due to different number of replicates used in the experiment. Sure, it's true that, so subgenomic N, if you're measuring it, it is a good proxy for the amount of virus that either is there or was there. Subgenomic N is by far the most produced by the virus. It is the #1 transcript that's made by the virus by a long shot, which is why we use that as our thing to detect it. They're asking us about replicates, but overall, my whole point of that statement was that the hamster does turn out to be an incredibly robust model where we don't actually see a lot of variation from experiment to experiment. And so this is very much what you were just looking at. So on the top, that's again, measuring the amount of N you're getting in the lung over a 14-day time course. But on the bottom, it's the same animals, the same kind of slides, except here we do staining called H&E. And this is really for physicians and pathologists where you can really tell what's happening in the lung thereafter because all the different immune cells stain slightly differently with this stain. And all I really want you to gather from this, though, is that there should be white spaces. You see on mock in day 2, there's lots of like little tunnels beside there that are just white, unstained, right? And those are air pockets where your air needs to go in order for you to get oxygen and exchange it for CO2. But you can see on day 6 and day 8, well after the peak of infection, which was day 4 above, you can see that those air spaces are largely gone and you're now packed with just pinkness. And we call that infiltration. So that is if you remember the call for reinforcements, that we brought in all these professional immune cells and they're coming to the lung because NF-kappa B has been on this whole time. And there's been a lot of chemokines being produced. And so all the immune cells are now coming to the lung to try and clean up this debris and deal with this infection. And by day 14 in the most hamsters, the infection is resolving itself and the animal gets better and recovers, very much like the vast majority of people with SARS-CoV-2. Next slide. Okay. If we take just a slightly deeper dive, this is just a couple slides here. If we go to 100x power, so we zoom in a little bit at what's going on in the lungs, there's a number of things that are interesting. And so the first one to mention here is what we call edema. So this is very much like when you saw the ground glass opacity, the clouding of the lungs. It's leakage. And so here, what you're looking at to the left, the bigger hole is your large airway, and then the little hole beside it where the red arrow is, that is a blood vessel, right? So obviously, when you're doing oxygen CO2 exchange, you need to be providing those red blood cells, oxygenated red blood cells, right to the blood. So those two things often run parallel to each other in the lungs. But there shouldn't be that like weird pinky fluid outside of the airway where the red arrow is pointing. And that's edema. That is a sign that it's leaking, and it's usually leaking because of inflammation caused by the virus. And so what's interesting about this is that it's showing you that you get leakage even where you don't have active virus replication, which is why the entire lung becomes very cloudy. Next slide. And if you zoom even further, now we're right up into the large airways. The other thing to note here is that at this moment in time, this is 3 days post-infection, now all of the cells that had ACE2 that were involved in your airways, they're all dead. The virus has infected them all and killed the vast majority of them. And now what's happening is you're getting this massive infiltration of a different type of cell called neutrophils. And the neutrophils are lining up where there should have been airway cells, and they're just trying to basically deal with the mess. They're trying to clean up all of the debris left behind, and they're trying to essentially squash this inflammation and get rid of the virus, before the virus completely wins out here. And so this is really probably the worst part of the initial virus infection, and probably really when you start coughing and having a difficult time being short of breath. Next slide. And so if you zoom in even farther here, what you can actually see is evidence of these cells dying. And so I just wanted you to appreciate that, like, this is not always programmed cell death or apoptosis is labeled here, but this is really like every type of cell death. So there's different ways for cells to die, and the virus basically triggers every possible type of death. If you get infected with this virus, that cell that was infected is basically as good as dead within 24 hours in some gruesome way. And you can even here see a lot of the red blood cells and stuff that are leaking into places where they're not supposed to be as a result of all of this damage and inflammation that has been caused. Next slide. Okay, so one of the really interesting things about this is that if you look systemically what's going on, so to the left here, that graph you see, that's where we can grab a tissue and look for infectious virus. And long story short is you find lots of virus in the lung, lots of virus in the olfactory bulb, but everywhere else you find very little virus. And so the measurements are here, like these are out of animals. And so 8 out of 8 animals in the trachea have virus, 8 out of 8 animals have virus in the lung. But when you look at other things like the brain, the GI tract, the pancreas, the kidney, it's much less consistent. It seems very stochastic as to whether or not a virus appears there or not. However, if you go to the next slide, if we take those snapshots that we were telling you about and we look for viral RNA, their viral RNA is more ubiquitous. You see viral RNA in tissues where you did not see virus. And that, again, really speaks to the biology that underlies COVID. So if you go to the next slide, if you go even further on that snapshot and you say, what's happening in all these tissues? Okay, so these are your pancreas, your GI tract, your liver, your olfactory bulb, your spleen, your brain, your heart, your lung, and your kidney. Out of all of these, only your olfactory bulb and your lung actually have replicating virus in them. But here, every little square you see that's red is a sign that you're turning on your antiviral defenses. And so the major take-home point here is that even though this infection is largely confined to your airways, your entire body is responding by eliciting this antiviral interferon response. And the reason it's doing that is because this virus is making such a mess that that debris from that mess is entering into your circulation, and it's triggering this kind of response in all of your tissues. So of course, if you are an individual that already had a kidney problem or already had a heart problem, getting SARS-CoV-2 is going to stress out that organ and push it. And so if it has any weakness whatsoever, this is when those comorbidities really come back to haunt you. Next slide. Okay, so in the interest of time, I'm just going to skip a couple slides. So let's go to the next slide. Yeah, we'll just skip this one. Yeah, we'll skip this one. Skip this one. I'll tell you what this all meant in a second. Just go to the next one, because I want to get to that. All right, so those slides were just really to say that the reason why we see systemic inflammation is because the virus is a slob, if you remember. Basically, we get debris from that infection, gets into the circulation, and everybody responds to it. All right, I want to get onto this, this other work. So while that's all happening in your overall body, I also mentioned that in addition to the lungs, your olfactory bulb does have active replication, which is unusual. So if we go back to the nose and go to the next slide, let's see what's happening in the nose. And so this is right back to that single cell sequencing, but this is a really cool experiment, just got published. And so we're looking at hamsters here, mock uninfected hamsters, hamsters infected at 1 day post-infection, hamsters infected at 3 days post-infection, and hamsters at 10 days where the virus is gone. And again, we're just taking a quick snapshot of individual cells, but we can use that snapshot of RNA to be able to figure out what kind of cells they are, whether or not they have virus, and whether or not they're responding to virus, for example. And so you can see the different types of cells that you have in your olfaction system on just a regular given day. And if you go to the next slide, we're going to remember these shapes. But now what we're going to do is we're going to color in the things we're interested in. So let's just look at the top for now. We're going to color in SARS-CoV-2N. So basically in that snapshot, wherever we see a piece of RNA that comes from virus, we're going to circle that cell blue. And so what you hope you can appreciate is that at 1 day post-infection, all the blue dots are really kind of down at the bottom of that shape in a variety of spaces. And then on day 3, those same blue dots kind of disappear in that space, but they go way up to the top at 3 days post-infection, and at 10 days, they're gone. So I'm sorry, but if you just go back 1 slide for 1 minute, that corresponds to, if you look on 1 day post-infection, there's an orange blob called sus cells, which are really just like epithelial cells that have this very much a structural role. And what's happening is that they get infected by virus, and the virus kills those cells. And so if you look between 1 day and 3 days post-infection, the population of sus cells drastically drops. Now that's not such a terrible thing because sus cells are just really structural. They're not the neurons that are causing you to smell. That's in the olfactory there. You can see olfactory neurons nearby, but they're not actually the ones involved in the infection. And what's interesting is, again, if you remember at 3 days post-infection, the blue signal went up. Well, those are all of your immune cells. You can see them as microglia and macrophages here labeled on 3 days post-infection. So again, advance 1 slide further. And so SARS basically comes in, infects those sus cells. The sus cells die. They make so much debris and such a mess that all of our professional janitors, the microglia and the macrophages, have to pick up all that debris. And that's why now you see that blue signal in those cells at 3 days post-infection. The graph below that is called ISG15. This is just one particular gene of that call to arms that we just talked about, the interferon response. And you can appreciate that, like, even though the infection is only in a few cells, the response to that, again, because it makes so much debris, is really, really strong. And as a result of that very strong response, your neurons that are normally involved in expressing receptors that can allow you to smell, their bandwidth is suddenly taken up by building up their antiviral defenses. And as they're building up their antiviral defenses, they no longer have the bandwidth to keep up with smell, and that's why you lose your smell. And the reason you get it back is that by 10 days post-infection, this eventually all goes back down to baseline and resets. And that's the beauty of your smell hopefully coming back. Next slide. And so you can zoom in on this and see that your smell disappears. So this ADCY3, that's a snapshot of a gene that's involved in smell. And you can see that in animals that get infected, that message goes away. And that's really the only point here, is that this is why you can't smell after your nose gets infected. Next slide. And so this happens in hamsters too. So I mean, in people and in hamsters, and so we can test this. So in fact, we just take Cocoa Krispies and we bury them underneath their bedding, and we determine how long it takes for a hamster to find it, because obviously it can't see the Cocoa Krispies, so it has to smell them out. And for this, we benchmark everything against an influenza infection, just because we want to know what are attributes of just, you know, being infected by a virus versus what are attributes that SARS-CoV-2 actually causes this. And, you know, long story short, very much like in people, hamsters get a nausea or a lack of smell, but eventually it comes back and everybody's fine. Next slide. And so how does it finally resolve? Like, that's obviously an important question. So if you go to the next slide, again, very much like us, what we see is, so onto the left here is just weight change. So when you get hamsters, hamsters really love to eat. And so if you just give them like a sham virus or some kind of mock treatment, they basically just continue to gain weight forever as they live in the animal facility happy. However, if you give them SARS-CoV-2, they feel less great. Obviously, they don't. They have this respiratory distress, and they don't actually lose weight per se, but they basically stop eating because they don't feel great, and so they just plateau. However, you can see that by day 7 to the right there, those are antibodies that are being made to this virus. And so very much like the average healthy individual of young age, when you get this virus, eventually your immune system figures out what it is, remembers it, and then, you know, works to neutralize it through the production of antibodies. Next slide. And so, you know, you can actually get really into this. And so this is actually where GenScript really came in very helpful, in that you can make different peptides to understand at a very, you know, way deep in the weeds of how the immune cells recognize virus. And this really comes in these two different forms. So B cells make the antibodies that many of us hear about after, you know, a vaccine, but T cells also play a very important role in that they can recognize infected cells, you know, very early on and make sure they get rid of those cells so the virus can't propagate. And this work here was really just showing the contribution that both B cells and T cells are very much involved in this process to ensure that you can get rid of virus. And so when things like Omicron show up and people panic because B cells, you know, aren't necessarily protecting against Omicron based on the way they were against the original Wuhan strain, it's not always as dire as people make it sound, because you're not actually measuring a whole aspect of the immune response, which is coming from T cells. And that's just because it's much harder to measure T cells. Next slide. All right, so here's the two models we're going to quickly test, and we're going to use this to really get into long COVID. And so we take SARS-CoV-2, we infect an animal, and what you're looking at here, these are just 4 animals on day 1, 3, 5, 7, and 14, and you're looking at the total amount of virus that's in their lungs at those particular times. And again, you can see all 4 animals track very well. The infection basically is producing a lot of virus for the first 5 days, then that immune system kicks in, and by 2 weeks out, there is no more virus left. The one on the right is the exact same animal, it's hamsters, but this is an influenza infection. So it's also an RNA virus, it's also in the respiratory tract, but a totally different family of viruses. This is actually swine flu, the pandemic we had in 2009, which was much more mild in comparison. And it has a very similar biology. Actually, the hamsters deal with that a little bit faster. But again, by day 3, they have very similar viral loads between the two viruses. And by day 14, the virus is gone. And those are going to be the 2 time points we initially focus on. Next slide. All right, so let's take a snapshot again and say what's happening into these animals. And so on the top, those are your lungs, and on the bottom, they're your olfactory bulb. And so if you remember, so basically on this one, blue means that genes are going up, and pink means they're going down. And it doesn't really matter what these genes are, but these are really depicting the aspects of your antiviral defense. And so, just quickly, you take flu or SARS, you infect animals, and on day 3, you see many of the same genes turn on. So all these blue genes that are turning on, these are all of your antiviral defenses. And by 31 days after infection, so a whole month after infection, your lungs are pink and healthy again, and everybody's fine. And so that happens regardless of whether or not you're infected with flu or SARS and you're young and healthy. Where the scene changes is the step below that in the olfactory bulb. And so that's the one we were just looking at the single cell level. And there on day 3, again, you see that both flu and SARS induce a response, but you can appreciate that SARS is inducing a bigger response. It's a darker blue. And that's because, again, remember, it's a sloppier, messier virus, so it's going to elicit more of a signal. But what's really surprising and really unique is that when you go out to day 31, a month after the fact, now you can see that even though with flu, everything went back down to baseline like you would have expected it to, for the olfactory bulb, SARS-CoV-2 looks like it's still infected even though there's no virus there. And this is was really the rationale for why we kept digging to see if there was long COVID there, because this is unusual. Everywhere else in the hamster, all the other organs that were not anywhere near the brain all go back to baseline on day 31. This is the exception to the rule. The olfactory bulb does this. And so we went deeper into the brain after seeing this. Next slide. And so just to really quick recap, basically, if you look closely, you can find the virus replicates very well in those sus cells, if you remember correctly. It's very easy to detect them by protein and by RNA in the lung on day 3. But at day 31, no matter how hard you look, you will not find any virus, no infectious units, no protein, no RNA. There's nothing left there, yet the tissue is behaving like it's still infected. And this is a really important part of the message I'm trying to convey in the remaining 8 minutes that I have here. Next slide. All right, so what we're going to do is we're going to go deep into the brain. So this was in close help with some neurobiologists and behavioral biologists and an amazing MD PhD student in my lab named Justin Frere. And basically what they did was they isolated very specific regions of different hamsters. So cerebellum, prefrontal cortex, the olfactory bulb, the striatum, thalamus, and the trigeminal ganglia. And they basically said, what is happening to these different areas of the brain? Are they infected by virus? Are they not going back down to baseline? What's happening here? And so you can find, again, pieces of RNA virus in those cells, very much like you could when we went and looked all over the body of the hamster or of cadavers from COVID individuals. But there's no evidence that there's actually replication competent virus there, just kind of inflammatory material, presumably from the dead and dying sus cells or something similar to those that have caused this. Next slide. And so, what's a little scary is this is just a slightly different way to show it. So here, what you're looking at is every gene in blue or red is a significant gene that's going either up, meaning it's bending to the right, or going down, which means it's bending to the left. And they're all very significant, meaning that we've done this in many animals, and the results are very reproducible. And you're looking at day 3 and day 31 at all these different areas. And so you can appreciate that, like, it's not uniform. So on day 3, for example, the prefrontal cortex and the thalamus show no activity at all. They look totally healthy and fine, just like the controls. Which is also true for day 31 for the prefrontal cortex. Whereas others like the striatum, you see lots of genes going up and down in both directions on both day 3 and 31, which is true for the olfactory bulb, the thalamus, both. We're seeing transcriptional changes that are not being resolved like they should be. And when you take a deep dive into those really do involve a lot of things regarding metabolism, like RNA metabolism and energy and inflammation, which are also many of the same triggers that we see in transcriptional snapshots of, say, Alzheimer's patients or ALS patients. It does suggest a certain amount of inflammation and problem in these very deep neuronal circuits. And so if you go to the next slide, this actually does play out in that when we do tests on these animals, the best one being the one on the left there, which is the marble bearing test. So what you do here is you basically take a hamster cage and you put marbles into the cage, and you basically measure how long it takes them to bury the marbles. And in mice, this is usually viewed as like a measurement of anxiety. And none of this has actually been done in hamsters because it's a relatively new model. So I don't want to over-interpret our results. But what we are comparing is mock-treated animals to animals infected with flu and then recovered versus animals infected with SARS-CoV-2 and then recovered. And all I really want you to appreciate is that while there was no significant difference between mock and flu animals to the point where we could actually group them together in this way, SARS-CoV-2 infected animals are performing in a way that would suggest that many of them have now abnormal behaviors. And so with marble bearing, for example, if you recover from SARS-CoV-2, those animals were just far less interested in bearing marbles than their controlled counterparts. time to approach is you put food in the light and they're in the dark, and you measure how long it takes for them to overcome their fear to get it. Whereas the one on the right, the time to first immobility, is basically a swim test where you measure how long it takes them to give up. And in all these cases, our SARS-CoV-2 recovered animals are showing abnormal behaviors from what they should be showing, suggesting that very much like people, there is a long-lasting effect that hangs on well post-viral clearance. Next slide. And so that I did make it to the end before the hour. And so the real major take-home messages here, I think, are that, one, I think SARS-CoV-2, it came from the wild. It was not created in a lab. It came from the wild, and its biology is such that it makes quite a bit of a molecular mess, and that mess of RNA causes inflammation in the entire body, including the brain. And what's really interesting about that is that while the body resets itself very well after that inflammation, the brain takes much longer to do that, and that really is the molecular basis for both why COVID hurts older people and those with comorbidities because of the systemic inflammation, as well as being the underlying factor for how it can cause long COVID, because obviously you are changing the transcriptional signaling of different areas of your brain to alter totally stochastic different aspects of behavior or memory, which is why the disease is so heterogeneous in nature. And so with that, I go to the next slide. I would be remiss if I didn't thank my people and our funding foundations. The vast majority of this work was funded actually by DARPA, which is part of the Department of Defense. The Ziegler Family Foundation, the Haas Family Foundation have provided amazing support as well. This work is actually accumulation of many people and many collaborations, far too many to talk about at this moment, other than to say Justin Frere did do a lot of this long COVID stuff. He's very active on Twitter, so if you want to learn more, certainly should engage him. But with that, if there's time, I'll answer questions, but I'm going to give it back to Sonia Hunt and say thank you for listening and attending.
Wow, you got through a lot. I think you did take a breath in there someplace.
Somewhere.
Well, thank you very much for that, Benjamin. It was very insightful and very detailed, and I hope the audience members really enjoyed it. Now we're going to try to see if we can get through some of the questions that have come through. Remember, audience members, you can send in your questions by using that chat box, and if we can attend to them, the team at GenScript will follow up with you. Now, Benjamin, before I start with the questions that just came in, did you want to answer any of the previous ones that I sent you, or just start with the ones that I just received?
Let's see. I will try and answer. Why don't you just ask me a question and I'll see.
Okay, no worries. Okay, no worries. Okay, this audience member just sent in and they're asking, the scientific community is divided about the underlying cause of systemic type 1 interferon signaling. How are you sure that there is no virus remaining in the animal after 7 to 10 days?
Right, so it's a great question. It's a really good question because obviously, the fact that you can get long COVID would suggest that it's really one of two possibilities. Either there is something that has fundamentally changed that is self-propagating and not resolving itself to cause long COVID, which is what I'm proposing. But you could also envision that there is a virus that's still present and inducing damage at some low level. And the problem with that is that it's obviously, if it doesn't exist, it's very hard, if not impossible, to prove something doesn't exist. And so what we do in this space, it's a bit tricky because if you are a human individual who gets hospitalized and unfortunately succumbs to infection, those individuals, that kind of tissue, before it would ever come to a lab that could be able to test for infectivity, is almost always fixed to make it safe for transport. But that process would kill any infectivity, and so really all you can do for the most part with human biopsy samples or human cadaver samples is to just check that snapshot kind of technique where you look for a protein or for an RNA. And that has been done, and you can find evidence of it. We also see evidence of that, of RNA in the brain or RNA in tissues where we don't think replication actually occurs. And so it creates a very quick controversy by saying, well, how did that material get there? Is it replicating virus or is it just debris? And so what we do is, this is where the hamsters again shine, in that if you if you can accept the fact that the hamster is just a small person, and you give them SARS and you then measure virus everywhere, we can take each individual organ or brain compartment, and you squish them to take out any kind of liquid or fluid that's inside of them, and you can take that fluid directly and try and look for infectious virus. That's the most traditional way to look for infectivity. But a second way to do this, which is even more sensitive, is to take that crushed tissue, or we call it homogenized tissue, and you literally add it on to like super permissive cells. So there are cell lines that we use in the lab that live in petri dishes from like African green monkey cells, for example, that are engineered to just be, it's how we grow the virus, for example. So they've been stripped of all their defenses. So just easy pickings for a virus. And so you would envision that, like, even if there was a single infectious particle in a given tissue, under that kind of experimental platform, the virus should grow out, and that would allow you to say, okay, there was infectious virus there. And very few people actually have the ability to do that experiment because it requires a very high containment facility and would have to be like tied into an autopsy, et cetera, et cetera. And so in hamsters, obviously, we can do this all in one pipeline without any disruption whatsoever. And when we do that, that's kind of the data I was showing there, that, you know, we don't find infectious material in the brain ever. We do find infectious material in the olfactory bulb and in the lungs, and then everywhere else, it's just like, it's very sporadic. So if you did this to 10 hamsters, you know, out of 10 hamsters, you know, one hamster out of the 10, we might be able to pull some replicating competent virus out of the heart, for example. And that doesn't necessarily, and that might represent true, in that it might be that, actually some of the work I skipped over because it was clear I wasn't going to have enough time, is that when the virus is replicating the lungs and it's inducing all of this debris and damage, a lot of that material that's getting into your circulation is causing that system-wide inflammation that I was speaking about. And while that's obviously bad, you know, it's a hard thing to go through to push all of your organs into this inflammatory process. It also does some good in that if the virus were to ever get out of the lungs and into the bloodstream or into the interstitial fluid of your body, if it gets to your heart, and it can infect your heart, but your heart is already inflamed because of all the debris that happened a couple days prior, it won't actually infect your heart. And so some of the data I skipped actually proves that point. It was done in Belgium. But if you take a hamster, for example, and you short circuit its ability to communicate in that way, then the virus goes everywhere. So it's almost like the systemic inflammation that the virus induces actually limits itself, in that it causes so much inflammation everywhere where it's not, it can't go anywhere anymore, because every time it tries to break into a new tissue, that tissue is so inflamed that it can't make ground there. And so this like sporadic sighting of a virus in a heart here or a pancreas there, you know, it might be real. It could be stochastic situations where, you know, people do see some limited virus replication in certain tissues, but it is not the norm. And I don't think it is the molecular basis of long COVID. I don't think we see any kind of active replication in the brain, and I don't think we're going to in the future.
Okay, I'm going to squeeze in one last question. This is a very interesting one. This audience member is asking, the title suggested you were going to talk about the past, present, and future events of the pandemic. You spoke about the past and the present, but not about the future. What do you think happens after Omicron?
Yeah, I did not come up with the title, and I was going to speak about the future, but you know, it's a fool's game, really. But you know, I have become quite optimistic in my old age, and you know, I know that many of my virology counterparts might not agree to this answer, but I really do think that the virus is running out of runway space. That you can only mutate your spike so much without compromising, you know, other aspects of its biology. And so I would like to think that Omicron is actually our last one for a good long while, and we should have, you know, a seroconversion in the planet to a level where we can probably keep it, and it will probably emerge here and there periodically as Omicron or Delta or some, you know, subvariant of those two. But I do think that the entire pandemic takes a major turn for the better here, and we go back to normal. But that's just my opinion.
Okay, well, thank you very much, audience members, for those questions. We have reached the end of the question and answer portion of the webinar. I know there's a lot of questions there that we were not able to answer, but please utilize the email addresses that are on your screen, and I'm sure the team at GenScript or Benjamin would be very happy to address them.
For sure.
So Thank you. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now give me a few seconds. I'm going to send you a link in your chat box. You'll be able to view the recording of this event at that link and also share this link with your colleagues once they register for the recording as well. So I encourage you to do that. And now let's please thank our speaker, Benjamin, for that very insightful presentation and for answering your questions. So thank you, Benjamin.
My pleasure. Thank you for having me, Sonia.
Okay, we hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Thank you again. Thank you, Benjamin. Thank you, everyone. Thank you again. All right, bye.